Hello friends, I am Neha Antal, Department of Biosciences, IMS Ghaziabad, University Coastal Campus. We already have discussed the external structure of prokaryotic cell envelope of prokaryotic cell. Now, the last part is internal structure of prokaryotic cell. Internal structure means which are consists of some granules, some liquid aqueous material, etc. Now, let's start. Cell cytoplasm. This blue structure is cell cytoplasm, which is aqueous in nature, watery in nature. The water content here it is a 70 to 80%. Gel matrix of water consists of enzymes, nutrients, waste, and other gases. It is also known as protoplasm. It is important. Cytoplasm is important as many biochemical reactions occurs here because here mitochondria and other organelles are not present. Site for many biochemical reactions and synthetic activities like location of growth, metabolism, and application. All functions happens here, occurs here. Also contain larger discrete cell masses like chromatin body, ribosomes, granules, inclusions, acting strands, etc. Now comes prokaryotic genetics. Genetics, gene, genetic material. What type of genetic material is present in prokaryotic cell? First come nucleoid. What do you mean by nucleoid, and what is the basic difference between nucleus and nucleoid? Nucleus is a well-defined structure, whereas nucleoid is not. The region of cytoplasm where chromatin material is present, that region is known as nucleoid. Usually a single circular chromosome is present. The basic genetic material is in the form of a DNA. Another part, another thing is present is plasmid. Plasmid is also a part of DNA, but this is a small piece of chromosome or genetic material, not critically to everyday functions, can provide genetic information to promote antibiotic resistance, virulence factors. Virulence factors, what do you mean by virulence factor? I have also already told you. The ability to cause the disease, degree of disease is known as कि कोई बैक्टीरिया कितनी डिजीज कोज कर सकता है वो उसका वायलेंस फैक्टर होता है. Molecules produced by a pathogen that specifically influence host function to allow the pathogen to thrive. Promote conjugation. Transfer of conjugation is a method of a sexual reproduction where both pilus comes in contact with each other, and one bacterial plasmid transfer its plasmid to another bacterial cell with the help of pilus, which forms a conjugation tube. The transfer of genetic material between bacteria through cell to cell contact. Now comes ribosome. Ribosomes, there are two types of ribosomes you have studied, 70S ribosome and 80S ribosome. 70S ribosomes are present in prokaryotic cell and 80S ribosomes are present in eukaryotic cell. 70S ribosomes present in prokaryotic cell, which are further divided into two subunits, one larger one and next is smaller one. Larger one is 50S and smaller one is 30S unit, which are again subdivided into 23S, 16S, etc. These ribosomes you can found ribosomes within cytoplasm or either it is attached with plasma membrane. They are made up of protein and ribosomal RNA. We have discussed it composed of two subunits characterized by S, that is Sevenberg unit, and these play a role in transcription, and it is also known as protein factory because proteins are synthesized with the help of ribosome. Next one comes inclusions. Inclusion, also known as inclusion bodies. Some bacteria lay down nutrients in these inclusions during periods of nutrient abundance. Uh, so some bacteria back up their food when if there is abundant if there is lack of food or nutrients, they can use food from their inclusions. Serves as a storehouse when nutrients become depleted. Some enclosed condense energy-rich organic substances. Some aquatic bacterial inclusion include gas cycles to produce or to provide buoyancy and flotation to bacteria. Here in this figure, see in this figure, there are poly beta hydroxybutyric granules are present in bacterial cell. Next come granules. Granules are also a type of inclusion body, contains crystal of inorganic compounds, and these are not enclosed by any kind of membrane. Example: sulfur granules of photosynthetic bacteria, polyphosphate photosynthetic bacteria, sulfur granules. In Corynebacterium and Mycobacterium, polyphosphate granules are present, which are also called metachromatic granules because they contain a contrasting color and methylene blue dye. Magnetotactic bacteria contain granules with iron oxide, give magnetic properties to the cell. Next come prokaryote endospores. Endospore we you can study as it's a different structure of bacteria which not all bacteria possesses or forms, and it's basically a structure when bacteria is present in inactive state. We studied we study reproduction of bacteria in three topics, three headings: vegetative reproduction, where budding, fission, and fragmentation comes. Second one, asexual reproduction. In asexual reproduction, we study this endospore formation, and in sexual reproduction, we study transformation, conjugation, and transaction. So basically, endospore. Endospore is a dormant, tough, non-reproductive structure produced by a small number of bacteria. You can see here it is a cell, bacterial cell, and you can see here endospores. This is endospore, and here bacterial cell is present. Endospore having tough cell membrane, which can hardly break down by normal environmental conditions. Resistant to radiations, desiccation, lysozyme, temperature, starvation, and chemical disinfectants. Endospores are commonly found in soil, water, where they may survive for very long periods of time. What now question arises: what are the two endospore producing bacterial genera that were introduced in our history of microbiology lecture? Search this and answer me in the comment box. Endospore generally we will study this on later slides. Next comes cytoskeleton. As our human body is also provided by skeleton to give a firm or solid shape to the body structure, like that cytoskeleton is present, which provides shape to bacteria. Cellular scaffolding or skeleton within the cytoplasm. You can see here these lines. These are known as cytoskeleton, providing firmness to the bacterial cell. Major advance in prokaryotic cell biology in the last decade has been discovery of the prokaryotic cytoskeleton, up until recently thought to be a feature of eukaryotic cells. And one more structure is present, which is known as mesosome. Mesosomes are generally the invagination of inner bacterial cell membrane which functions to reserve food. That's it. Thank you guys for listening.